Hi, my name is Michael Snyder. I'm a professor and chair of genetics at Stanford University, and I'm also the director for the Center for Genomics and Personalized Medicine at Stanford. So I'd like to tell you today about our work for using genomics for understanding human health and probing personalized medicine. So I'm going to start with the premise that all of you know, and that is, if we can go to this slide here, your health is a product of two things. One is your genome. Hello, sorry, having some technical background here. Okay, so once again, I'm Michael Snyder, and I'm going to tell you about our work in the area of genomics and personalized medicine. And basically, we're going to start with this premise on this slide. That is, your health is a product of your genome and environment. So at the top is the genome, and the bottom is the exposome. And these days, we can get at the top part of this, that is your genome, through the advances in DNA sequencing technologies. I think many of you probably know that actually a lot of what I'm going to say has really been precedented by a movie called Gattaca that many of you may have seen many years ago. The idea that your genome, along with the various things you might be exposed to, your occupations, what have you, will basically determine possible outcomes for your health state. We can now, as I say, get at the top part of this, your genome, through the advances in genome sequencing technologies. So starting in about 2004, 2005, there's been a revolution in DNA sequencing such that the cost of sequencing has dropped dramatically. It's about tenfold every 15 months or so. And it might be tailing off a little bit over the last year, but it is still going down, and there are new technologies coming out so that we expect prices to continue to drop. But nonetheless, even today, for a research genome, it costs about $3,000 to sequence a human genome, and it costs much, much more to analyze it, though. So the sequencing is no longer the limiting step or price limiting step for analyzing a genome. It's really the interpretation, and that's something you'll see as we go through today. But because the cost of genome sequencing has dropped so dramatically, it's had a huge impact on medicine and personalized health. And this next slide here actually shows a lot of different areas in which genomics is having an impact on medicine. First, it's being used to understand and treat certain diseases such as cancer, unsolved genetic diseases. I'm going to get into this more in a minute. It's also being used quite a bit for pharmacogenomics. These tend to be more specific tests, but you can actually get this out of a genome sequence. And a topic that interests us a lot, and that I'm going to spend some time, is really where this is all going in the future, which is using genomics to manage health of healthy people, not just those who already have a disease. So if we go to this next slide, as I say, genomics is having an impact in certain areas, and perhaps one of the most profound areas is, in fact, cancer genome sequencing. So I think most of you know that cancer is a genetic disease, both in terms of its risk for inheritance, as well as somatic changes that occur as people go on in life and acquire mutations. And it's thought that there's somewhere on the order of maybe 5 to 20 different driver mutations that are responsible for causing this cancer. That number is up in the air, and it probably varies a little bit with the different cancers that we're talking about. But nonetheless, there are multiple mutations that are responsible for genetic mutations that cause a cancer phenotype, that is, for cells to grow uncontrollably. And so the idea is that one will go in and sequence genomes both from the cancer tissue and from normal tissue, and try and find genetic changes that are responsible for for these cancer phenotypes. And then ideally, you try and find changes for which it might suggest therapeutic treatments. And so we've done this, as many other groups have, for quite a bit. It was probably led a lot, in part by Bert Vogelstein's group, Elaine Martis and Rick Wilson, the latter at Washington University. But we've also done quite a bit of this, and I can tell you that in virtually every case we get information that is useful for treating a disease. And one such case is shown on this next slide here. This is actually one we just did fairly recently. This is a patient with metastatic colon cancer, so very late stage cancer. And just by sequencing the genome, one could see two regions that jumped right out at you. There's an amplified region for the EGF receptor gene, which is generally not very common in colon cancer. It actually only happens about 2% of the time. But because this individual had this kind of mutation, and also because there are drugs on the market for EGF receptor, there are two drugs, in fact, already approved for this, not for colon cancer. They've been approved for other cases, but it's clear that it's an obvious candidate to use then for therapeutic treatment of this individual, and in fact we are treating this individual with one of those two drugs. So the point is that actually you can get changes suggested that will lead to treatments, and as I say, we find information that both tells us what we probably should be treating with, as well as information that what we might normally treat with may not be useful. So negative information is sometimes valuable as well. And all of this is happening because different cancers have different genes mutated. In fact, it's going to be common to classify genes more by the mutations rather than their site of origin. So this is more informative to call this patient as amplified for EGF receptor rather than colon cancer, because that's really the relevant thing that's going on. And so one lesson out of all of this is that it's going to be clear that this is an avenue available to people for treating this and for treating cancer. And I think all of us expect that this will become standard of care probably within a few years. This isn't going to be 20 years away. This will be coming in the very near future. The other area where genomics is having enormous impact is in fact in issues of childhood diseases, Mendelian diseases, and sporadic diseases that appear. And there's been a number of cases that have tested and a few spectacular successes. I'll tell you right off the bat, most of the time this does not work, and I'll show you some of the challenges associated with this. But I think one of the more spectacular cases is a case that was done by Richard Gibbs and Jim Lupsky at Baylor, where they actually looked into a family that had this disease here. Turns out that there were two twins that had, it turns out, dystonia. They were born, and from a very early stage it was recognized there were problems. The babies were floppy and they had various abnormalities, and they were really quite sick for all the early years of their lives. And it was actually the mother who figured out that the children had dystonia, and they treated the children with L-dopamine, and that actually improved the health of the children. They weren't completely cured, but it helped them quite a bit. But they still had some problems, and then many years later, actually just a few years ago, they sequenced the genome of these two twins along with the parents and discovered that in fact they had a mutation in this gene with the red X. It's called the SPR gene. And that gene, it turns out, is involved in dopamine biosynthesis along with another very interesting neurotransmitter, serotonin. And so what they wound up doing was administering to the children not only dopamine, which they were getting already, but also serotonin, which in fact basically is a precursor serotonin they use, which basically cured the remaining symptoms. And so the children are very well managed these days, and the genome sequencing really led to a very useful treatment that dramatically improved the health of the children. So the bottom line is this is one of the more spectacular cases. So we've taken on, I'd say, about 30 of these cases so far now. We have had success, I would say, in about 5 cases. I'll just show you one here. In none of our cases have they led to treatments yet, I'll have to say. But in one case shown here, this is a collaborative study with a variety of investigators at Harvard and other institutions. And basically, these are individuals with a combined immunodeficiency, but this is a very special type of combined immunodeficiency. These individuals also have multiple intestinal Atresias. And it's a very rare disease. As I say, they're intestinal problems, and they also have immune problems. And so we were fortunate enough to have 5 different families, as well as several other individuals who were affected by the disease. The DNA was available to us. And so we performed exome sequencing, just sequencing the genes of these individuals, the gene coding regions. we wound up sequencing 5 affected individuals. They're the ones shown with the black boxes or squares or round, black round circles. We also sequenced a number of unaffected individuals because that's very informative as well. And so you sequence these family members and then you try and find mutations that would be consistent with individuals having the disease. So if they're black and if it's a homozygous mutation, they'll have 2 different mutations within the gene, either different mutations with the same mutation within both copies of the genes. And the unaffected would either have no mutations or 1 mutation if it's a parent. And so we sequenced 15 individuals, 5 of them were affected, roughly around the 100-fold coverage. And what we discovered is by comparing the results of all 5 families as shown here, it did take all 5 families in this case, we found 1 gene that was mutated in each case in the families that was, again, in each of the families, and there were no other variants that were there that were consistent with the segregation pattern. And this gene then turns out to be the TTCA7 gene. And by looking at this, we saw there were a number of different mutations in the gene that basically varied quite a bit with the individual families. But as shown on this slide, F2 means a family individual, F3 another family. And basically you can see the mutations are scattered at different locations in the gene. Each of these was predicted to be deleterious by algorithms that tell you about this sort of thing. And the bottom line is we could pinpoint this gene as responsible. Now, one thing we learned from this, and now many other studies we've been carrying out, it's probably more common than not that the mutations turn out to be what are called compound heterozygotes, where there'll be 2 different mutations, one in the mother's allele, one in the father's allele, but they're different, and so each is inactivating and will disrupt the gene function. And this is very, very common in many of the families that we've analyzed. So this is one of the 5 successful stories I can tell you about. As I say, in this particular case, it didn't help us therapeutically. Here's a case that's more of a very common scenario where we've taken on families that have hereditary hearing loss to see if we could understand the underlying genetic defects of these individuals. And there have been a lot of genes identified in this, and that's one of the problems. There's actually almost too many genes. There's, in fact, about 64 genes that have been identified associated with hereditary hearing loss so far, and it's clear that we're not done yet. So we went in and sequenced, in this case, 3 different families, did exome sequencing of 3 different families shown here, and we also sequenced a lot of individuals who had hereditary hearing loss as well, at least as far as we could tell. But we only had 1 affected individual. And we were just curious how many times we could solve the problem, that is, understand what the underlying genetic defect was by sequencing these various families and these individuals. And to make a long story short, we actually sequenced all of the family members in each of these 3 families and all of these 13 other individuals to see what we might be able to see. And I can tell you, only in 1 of the 3 families were we able to identify what we think is the underlying genetic defect. This is the case of family 1, where by sequencing these 5 individuals, there was 1 defect that was consistent with the segregation patterns. That was in the MY7B gene, encoding a myosin heavy chain, which is fairly similar to another hearing loss gene that had been identified, another myosin heavy chain. So we think this is the causative mutation in this case. In family 2, which is a very typical scenario, we got a short list of several genes that were consistent with the segregation patterns. We don't know which one's right yet, which one's wrong. So we have to sort through those candidates by other tests. And in the 3rd family, the list is just very enormous, and so we don't know which ones are responsible. And I can tell you, just getting to the end of the story, that the individual probands that we sequenced, the individuals, in all cases, was not informative for telling what the underlying genetic defect is. This is just too complex a disease. And so these are just some of the kinds of problems you run into when you're doing these sorts of studies. And so really, to figure out the genetic disease, these are some of the things you really have to understand. One is that you're best off solving the disease if there's already another individual who has a mutation in that same gene. And we've now found this in several cases, where these so-called recurrent mutations are really the best way to pinpoint the likely gene responsible for the phenotype. So you either have to look through many families or by looking through the literature of the database. In some cases, we've looked through blogs to actually find a recurrent mutation, and that's been very informative. So there's a lot of interest then in setting up common databases for being able to share this sort of information, because, and the families, of course, are very interested in this sort of thing, because at the end of the day, if you can find someone else with the same variant, at least a variant in the same gene that's predicted to be disruptive, you can solve these, solve what the underlying genetic cause may be. The other thing you can use, and we all do this, we look for similar phenotypes, the mouse models. There's a lot of data out there. There are other groups that are trying to recreate the phenotypes. Once you find county genes, you go in and quickly make mutations in model organisms like zebrafish to try and understand if you get a similar phenotype. Or you look for genes in similar biological pathways that might be defective in individuals with similar disease phenotypes as well. And so a classic case here is like the one I just said with the myosin heavy chain, because there are mutations in the cytoskeleton and other myosin heavy chain. It wasn't such a stretch that the myo7b gene was likely responsible. So there are a lot of challenges to sorting this out. This really is the bottleneck, trying to find which one is the cause of the mutation if you're not so lucky to have one mutation that really stands out. So those are using genomics to try and understand phenotypes of people who have disease already. But really where genomics is going is this concept here, the idea of using genomics to manage health of healthy people. So I imagine many of you who are listening in on this call may not have disease problems already, but you're kind of interested in how genomics might be useful to you. And so the underlying question is, if you were to get your genome sequenced, would that be useful to you? Could you learn something about it that might help you manage your health condition? And we're certainly believers that this is the direction the field is going to go. That is, people will get their genome sequenced, use this information to be on the lookout for diseases and such. And specifically, what we would envision is a scenario where you would use genomics, if you look at the top left, where in conjunction with your physician, you would use this information, looking now down to the bottom right, you'd use it to predict disease risk, maybe diagnose diseases early because you'd be on the lookout from your genomic information. If you had a disease, you'd use genomics to monitor disease states. You could certainly use genomics to help treat disease, suggest drugs like cancer I just mentioned. And finally, minimally, you should be able to use genomics to try and understand why you may have a certain disease state. And even if you can't do anything about it, it's still very useful in many cases to try and understand the underlying genetic basis of this disease. So we're very interested in rolling this general concept out, and we didn't just want to bring in genomics, we wanted to bring in other sorts of information. Specifically, we wanted to bring in other kinds of omics information. And this is reflective of the fact that I used to get a bit frustrated when I would go to the doctor's office and they would draw blood and they'd give you back a report that would show you about 15 different things they measured. And our lab is one of the few labs that does a lot of genomics and other sorts of omics, and I always wondered why we weren't measuring many more things. So we not only wanted to bring in genomic information, we wanted to bring in other sorts of information, transcriptomics, proteomics, metabolomics, and other sorts of information. And so many of you may know I was at Yale University for many years, and I moved to Stanford just very recently. And so basically, when I moved to Stanford about 4 years ago, we set up something we call Integrative Personal Omics Profiling, or iPOP. And the idea is that in addition to your genome at the top, you would do all these other omes on you on a blood test and get a much more detailed perspective of what was going on with your health state. And you wouldn't just do one time point, you'd actually roll this out over a series of time points so that you could follow in more detail really what was going on and how your health state might change. And specifically, just to go from top to bottom, we would not only look at the genome, we would look at the epigenome, in my case the DNA methylome, the transcriptome in your mRNA, your microRNAs, its various isoforms and edits. We'd look at the proteome, and in their case, it's really what you see using mass spectrometry. For the aficionados out there, we use something called TMT labeling to look for differential changes in the proteome. We follow about 4,000 proteins. And this is primarily targeted, I should say, at peripheral blood monocytes. So everything we're doing here is out of blood components, and now we're doing a little bit with urine, but mostly we're doing out of blood components because that conforms to the existing medical structure that people use and medical tests people use. And so we mostly do, for example, transcriptome and proteome and a lot of these things out of peripheral blood monocyte cells, but some things we do out of plasma or serum. So we follow the proteome by mass spectrometry. We also follow more rare things that you don't necessarily see by mass spectrometry. We follow cytokines, which are a very important class of molecules involved in mediating the immune response. We also follow metabolome, and in our case we do untargeted metabolomics, where we'll basically do mass spec profiling once again. We follow about 5,300 peaks, of which 1,000 we know what they are. The remainder, they're just peaks. We don't know what they are, but 1,000 is still pretty good compared to what's currently done for blood tests. We also follow autoantibody profile, which initially was following reactivity to 9,000 proteins. These days we follow reactivity to the whole proteome, along with actually the whole virome. We can see your reactivity in your sera to all these, all known viruses and all known human proteins, all on a chip-based assay. And lastly, most recently, we've been adding on the microbiome, where we're actually following gut, urine, skin, nasal, and tongue microbiome. So 5 different microbiomes where we're profiling DNA, and we're going to start profiling RNA and things as well. So we're basically following all these things. Initially, we simply followed all these things, but not the epigenome, because it was kind of expensive back then, or the microbiome, and we were following about 40,000 molecules initially in blood. These days, with the epigenome and the microbiome, we're literally making billions of measurements, and I'm not quite sure how you exactly count this in terms of, it's definitely billions of measurements. It's maybe not billions of molecules, but it's certainly very large numbers. And so you're doing billions of assays to really follow a person's health state, if you will. And so we initially rolled this out on me. Again, many of you may know the story, so I'll go out over kind of quickly. We did it because we wanted to have access to material, and we wanted to make sure we were getting samples when people weren't feeling so well, as you'll see in a minute. We also did it because at the time, genomics was a little bit controversy. A lot of people worried that people would learn things that they couldn't psychologically handle, like they may learn a risk for a disease, and they wouldn't be able to handle that. And so it just seemed a lot easier to start this as a proof of principle study with me, where I didn't have to worry about people getting upset. If anyone got upset, it would just be me, and that would be okay. So we rolled this out, as I say, on me. The study really started, we had a blood sample from when I first came to Stanford 4 years ago, and then actually when we launched the study was day 0. You'll see that in the top left. And it just so happens on that day I had a rhinovirus infection. So we took blood that day. 4 days later, if you look to the next notch to the right there, and then 21 days later we took another sample. And then we basically sample about every 2 to 3 months when I'm healthy. But it turns out I have little kids, so I pick up virus infections quite a bit. And in fact, I've now had 6 viral infections over the last 3 years. We've been collecting samples for about 3.5 years now. So 9 months after my first viral infection, I had a very nasty respiratory syncytial virus infection. And this is fairly typical. Literally, as soon as I was feeling congested, got a little sick, we took blood that day, the next day later, 2 days later, 2 days later, 3 days. You can see quite a few samplings. We do dense sampling when I'm sick, and then about every 2 to 3 months when I'm healthy. And as I say, so far over the last 3.5 years, I've had a total of 6 viral infections. There are 3 rhinovirus infections. You can see they're the ones in red. This RSV infection I mentioned before, and now 2 adenovirus infections. And the other thing you'll probably notice is that I typically seem to get sick about twice every winter, and that's how it's been popping up. So we basically have, as I say, collected samples from all these states, and we've been analyzing them. And I'm just going to summarize the results because I've presented this many times, and also we have published this, but I'll give you a few highlights, and then I want to talk about some lessons and future directions and new things that we've covered as well. So we started by sequencing my genome very deeply with several different technologies. I'm going to come back to this in a minute, but we generated a very accurate genome sequence by using different technologies, Illumina and Complete Genomics, along with several different exome sequencing technologies, and came up with a list of 3.3 million high-confidence single nucleotide variants, about 200,000 insertions and deletions that are small, and then about 3,000 structural variants, which are larger deletions, but they're not as many, but they do affect a lot of nucleotides. And then what we did next was we phased the genome. Now, it's interesting, in my case, we actually phased my genome initially by sequencing my mother, and that worked pretty well. But there's some new technology out there that gives you essentially long synthetic reads, if you will, by Moleculo, now part of Illumina, and they basically generate roughly 10 kb reads or so. And that's long enough you can capture several variants on the same synthetic read length. That is, over 10 kb, you'll get on the order of 6 to 10 different variants in that segment, and that's enough then to link up the different variants so that you can assign the variants to one chromosome or the other, or say your mother or your father. And that worked really, really well for us. In the end, we were able to assign 98.2% of my variants to my mom's or my dad's chromosomes using this technology. So as I say, it worked really, really well for us. What we did next, actually, or early on, the molecular technology is actually quite new, is we've analyzed the genome for medical risk. And we do two kinds of analyses here. We look for rare variants that are likely to be highly penetrant for causing human disease. And then we also do a more integrative analysis of the 23andMe sort, where we integrate over multiple markers for complex disease to look at disease risk. So this first slide here that I'm showing you actually shows a pipeline set up by you and Ashley at Toll Butte and Russ Altman to look at dealing disease risk, highly penetrant diseases. And I won't have time to go through a lot of this other than to say that we spend most of our time looking at the coding SNPs. This is a pipeline we set up and used early on. These days is a slightly revised version of this, but we spend most of our time looking at protein coding sequence variants. That's the pipeline on the right. We do look a little bit at the non-coding ones, mostly in splice regions and to some extent in microRNAs, but mostly in the splice junctions. And these days we're looking at regulatory sequences, I'll get to a little later on. But we spend most of our time looking at protein coding variants. We zoom in mostly on those that are non-synonymous, meaning they cause amino acid changes. And then we look at those that are predicted to be damaging by various algorithms. And so the net result is we then take those and we zoom in on those that are associated with human disease. So there's a database online, Mendelian Inheritance of Man, that has curated variants associated with human disease, and we zoom in on variants again that are associated with human disease that are predicted to be damaging and that are rare. And so I have 51 of those, as you'll notice in this box. And so going to this next slide, this is actually the list of those rare variants that are associated with human disease. And for the most part, mine actually looks pretty tame, pretty mild. I did see mutation in telomerase gene. That's this red box here associated with aplastic anemia. I'm going to come back to that in a little bit. And then we also saw mutations associated with other things. It was known going into the study that I had high cholesterol, and we think the mutations I have in this gene, GCKR at the bottom, are likely to be associated and causing that high cholesterol phenotype I have. I actually have two different mutations. Once again, a compound heterozygote, one damaging mutation in my father's allele, one in my mother's allele, that are probably responsible for my high cholesterol. I'm a great responder to statins, so we've got that under control. And then actually, not indicated here, but to the right or to the left of that GCKR gene are two other genes, KCJN11 and KLF4, that are actually associated with type 2 diabetes. We did notice those through this analysis. They're not rare alleles, but because these are common diseases, we did keep an eye on those. As I say, the other thing we did was we looked at a more complex disease analysis, and this is where you integrate over multiple markers. And Toll Butte has assembled a pretty amazing database called VeryMed that actually has collected a lot of information from the literature, and they've calculated likelihoods from all these genetic association studies. And then they integrate over the various risk and protective alleles to predict whether you're increased risk or decreased risk for hundreds of diseases. This slide just shows a few of those, maybe something like 10 or so different diseases, but they've actually looked through hundreds of diseases. And basically, to walk you through this slide, as a European male, I'm at the triangle is my starting point for being at risk for one of these diseases. And then on the right is 100% chance of getting the disease, and on the left is a 0% chance. The orange bar means my increased risk amount for one of these diseases, and the blue bar means my decreased risk amount. And the net result is that, for the most part, when we looked at this, they call this a riskogram. Most things made sense with my family history. So, for example, at the bottom, I'm at decreased risk for obesity, because, and that made sense, nobody in my family, going back to my great-grandparents, has ever been overweight, as far as I can tell. Also, if you look at other things like increased coronary artery disease, the bar is actually a little bit further than it looks here, but it's that made sense too, because everybody on my father's side has died of heart disease. You can see the increased lipids and cholesterol in two different places here. So I am in it, and that made sense with my high cholesterol. So all of that made sense from my family history, but there were some surprises, two in particular. One is I'm at increased risk for basal cell carcinoma. That was something that came up relatively recently, and I didn't know of that running in my family, but ironically, I did happen to talk to some of my other family members. I have 3 sisters and 2 brothers, and I actually discussed this with one sister and 2 brothers, and it turns out they've each had multiple lesions of basal cell carcinoma. So in fact, this is in my family history, I just didn't know it. So one of the arguments I like to make is that your genome is a better indicator of your family history than your family is, because your family doesn't always discuss these things in the open. And unless you have access to everybody's health records, you may not know what's exactly in your family history. So as I say, basal cell carcinoma is just not something we ever talked about at the dinner table. So it really never came up. Now, one thing that came up very early in the study is obviously this thing with the big arrow, where I was shown to be at increased risk for type 2 diabetes, where I went from something like a 25% chance to 47% chance of getting type 2 diabetes. That was also a surprise, not so evident from my family history. And so we did look at this further. Now, remember, we've been doing all this other omics profiling, so we were following my glucose. We're doing standard medical tests as well. Followed my glucose and virtually everything else you can imagine over these last 3.5 years, and this just shows you some of the information from the glucose part. And what you'll notice is, if you look on the left, my glucose is marching along just fine. It didn't matter whether it's fasting or not. They were always pretty much in the normal range, typically 90 when I'm fasting, or maybe up to 100 when I'm not, but it was nothing to be concerned about. But ironically, right around that day 320 or so, it was a day I went in for a glucose metabolism test, and I went to get this test because my genome basically suggested I was at risk for diabetes, and I wanted to see how my metabolism went. And so when even before we started the test, they measured my glucose, and you'll notice that's the one at that inflection point. My glucose was 127, which is high. That's not normal for fasting glucose. And we were both surprised, and so we repeated the test, and sure enough, it came out the same number. Went on, and my glucose metabolism seemed okay. But obviously, because it was high, we did a lot of follow-up tests, and you'll notice if you keep going to the right, a week later, my glucose increased to about 141 or so. We also measured hemoglobin A1C, which came out actually quite high, 6.4%, which is quite high. Diabetes is typically classified as 6.5, so it's right on the edge. And then about 4 weeks later, I went to my general practitioner, who also made all the measurements and then did the hemoglobin A1C and glucose, and came out even higher. So glucose was 150 then, hemoglobin A1C at 6.7, and so the net result was that it was pretty clear that after a period of time, my glucose and hemoglobin A1Cs were high, and in fact I was classified as diabetic. So I can tell you when that was. That was April 11th of 2011. It's just after that 6.7 is when I got the news back, and at that point I decided I had to do something. It was clearly prolonged up there for a time. It wasn't just a few days. By then it had already been a few months. And so basically I completely changed my diet. I pretty much had the world's worst diet, meaning I ate a lot of desserts and things pretty much every night, and I just essentially cut all that out as of April 11th, 2011, except for one bite of wedding cake I've had summer of that year. Otherwise, I really don't touch that anymore. And I also increased my exercise, and it didn't happen right away. You can see as you look off to the right, but gradually my glucose came down to normal, and for the most part it stayed down ever since. And I've been measuring, as I say, for 3.5 years, so this bar keeps going to the right. Bounced around a little bit last winter with some of the viral infections, but generally stayed pretty low. So the bottom line is that my genome predicted I was at risk for a disease, which in fact I did get. And so this is really the first time genomics have been used to do that. But because I did, in fact, follow this, because I had my genome sequence, I did catch it early, and I was able to manage the disease, which is nice. The other thing you'll notice is that this all appeared after this very nasty respiratory syncytial virus infection, which is very unusual amongst adults. It's quite common in kids, but not that common in adults. And I was actually out for several days. It was, you know, I was quite sick, and it's clear that that's when my glucose spiked just after that. And so our hypothesis is that my genome has me at risk for diabetes through the genetic risk alleles I have, and that in conjunction with this environmental stimulus, the RSV infection, is what's triggered the disease. Now, we can't prove that, but it certainly leads to a very strong hypothesis that you can follow up with other assays. And I can tell you I've gotten lots and lots of requests and comments from various people who have actually had similar experience. So anecdotally, I can tell you this is happening quite a bit. And that kind of connection had never been made before for type 2 diabetes, which is a form I have. It has been made for type 1, but not for type 2 diabetes. Okay, so the other thing I can tell you is that we've been measuring all kinds of things, cytokines, the transcriptome, proteome, metabolome, and I don't have time to go into this, but the bottom line is you can follow all of these molecules and pathways changing at a level no one's ever seen before, and many of these are very relevant to either viral infections during the time I get sick, or in the case during the time I got diabetes, you will in fact see very interesting pathways changing during those times. And this is just one example on this slide here, where we saw spikes of activity that occurred about 2 weeks after I got the RSV infection. This is an integrative analysis of transcriptome, proteome, and metabolome. It took quite a bit of a computational expertise to be able to systematically pull out the pathways that change as you go through these various disease periods. And we had a physicist, George Mias, who figured this out and set up a nice framework. And I forgot to say that Rui Chen is one of the spearheads of the study. I'll mention all the people at the end. But the bottom line is you can do things like this. You'll see this giant spike if you look at the top panel, that red bar that's there. Those are all the molecules that are going up at this 2-week period, and all kinds of interesting changes are occurring. Spliceosome changes, but one of the biggest pathways that goes up is, in fact, glucose regulation of insulin secretion actually is increased during this period. And that is primarily a proteomic signature, the yellow dots, if you look to the right, are the components that are changing, and those are primarily proteomics. And then there are other things that go down during that period. There's all kinds of signaling pathways that change. Interestingly, my platelets are actually, my platelet plug formation pathways going down. And this is actually a mixed response of transcriptome and proteome. You'll actually see some interesting metabolites change too. And that goes down. And why is this interesting? Well, this is the power of integrative omics profiling, because what you can do is you can see all kinds of pathways change at a level you wouldn't see if you only followed one ohm. These mixed signatures will only, they'll reveal pathways that appear, again, that you wouldn't necessarily follow had you only followed a single transcriptome. You would have missed both of these if you only followed transcriptome. By doing the proteome, and you can follow other pathways that change, and you need, for some cases, you need it all to see the various pathways that are changing. And it's really been incredibly informative to follow this. So we're a big believers that the future is going to be here. That is that, you know, in my world, I think I would be an advocate for people getting their genome sequence at birth, or quite frankly, before birth, with the technologies being where they are these days. We would also then combine other omics information and use that to actually, when you're getting your various health task when you go to the doctor, we would follow things at a much more detailed level than we're currently doing. I'm not saying you'd follow billions of things in the ultimate clinical test, but you'll be following many, many more things than we're currently doing. You also bring other information, as I'll get to in a minute, and you use this information to predict disease risk, diagnose disease early, and monitor and treat disease, all the things I said earlier. Now, I will tell you, I don't think that everyone necessarily needs access to the genome sequence. It should be up to them to decide if they want that information. But I do think that some of the information is valuable and could be relayed back even right at birth, much like PKU is relayed back at birth. And other information would be available as you needed it if symptoms arise, or if you saw you're at risk for certain things, you could actually use that information to try and keep an early look out for that, or even adjust your lifestyle. And ideally, you'd like to be able to get this information, predict what your risks for, Dizygotic, and then monitor your activities so that you don't put yourself at risk for those sorts of things. So that's the world that I would like to see in the future. Now, just to bring up a few things, people say, well, Mike, if you do these tests, you're going to wind up saying, if everybody gets their genome sequence, you may wind up saying, they have lots of disease variants, it's going to cost hundreds of thousands of dollars. They get all this information they can't use. The whole thing's a mess, and there's 100 reasons why you shouldn't do this. And so working with you and Ashley and Tom Kutermes, a group of us, of Rick Dewey and Megan Grove and Queeping Pond in particular, actually just went out and tested that assumption that people are making. And we asked, if you sequence the genomes of healthy people, how often will you find information that would be of use to a person that would go back to them, and how much would it cost, and what kinds of information might you learn from healthy people's genomes? And so what we did was we sequenced 5 Asians and 5 Europeans and basically looked through their genomes and looked at what kinds of reportable disease risk information might be there. There's been some guidelines set up by the American College of Medical Genetics that basically has 57 loci identified that they suggest you report information back to patients on. And so we looked at those 57 loci, and it turns out from our small study, which is 10 people, we looked at their variants actually in exquisite detail, so it's more than these 57 loci. But even from the 57 loci, we found a median of 5 reportable disease risk associations, including one individual who had a BRCA mutation, did not have a family history. So we would argue that's useful information. And the range you can see here is 2 to 6. From these 5 reportable disease risks, this led to 3 follow-up diagnostic tests. And the cost was somewhere on the order, you can see here, about $400 to $1,400. So, not dirt cheap, but not super expensive either. So somewhere in a reasonable range, and one would argue that this actually could save a lot of money in the long run. So the bottom line is this is very doable. You do get some useful information from virtually every person we sequenced, and in some cases we would argue it's extremely valuable. Now, it's not simple to do this. It takes about 54 minutes per variant, and if most individuals have somewhere on the order of 50 to 100 different variants that you look up, it does take quite a bit of time. We spend about 40 hours per genome to go through all the variants, not just the 57 loci. So it is quite a bit of effort to do this. So that part is up there. But nonetheless, we would argue this is the kind of information you might learn by sequencing a normal person's genome. Okay. Now, I won't tell you this is all a piece of cake. There are a lot of challenges associated with this. I've listed a few here. You can see the numbering got a little bit screwed up, but these are just some of the issues it's a smallish list. There are many more challenges here. These are just some of the things, and I thought I would go through a few of them. First of all, genome sequencing, accuracy, and coverage is still a big issue. People don't realize this, but getting your sequence doesn't mean you're getting a nice, perfect genome sequence. And we knew this early on when we sequenced my genome with complete genomics and Illumina, and only 89% of the single nucleotide variants overlap with these 2 technologies. Both work pretty well, and we could validate a lot of variants with both. The common variants are always right, and the platform-specific variants for complete genomics or Illumina are often right. They're not always right, but one platform often misses, another one finds. And these are often very interesting variants. Here's some of the validation data on this slide. So you can pour through these and get a sense of what the accuracy is. And I can also tell you that a lot of these variants are very interesting. Some of them have been associated with human disease. So you do want to collect the information from a lot of these loci. And this is just single nucleotide variants. For the indels, it's even worse. We only found 28% overlap of this, and so there's clearly a lot of issues with accuracy. And it has improved even since this first study, but there's still a long ways to go to get perfect genomes. This actually just highlights the result most recently. This is from a company I'm affiliated with, Personalis, where they actually sequenced the genome with the exact same DNA, but did it twice, and fairly deep, the 60x. And what you'll discover is that, first of all, there's pretty good concordance overall where they do agree, but you'll discover that one run will find things the other one run won't find. So for example, run 2 found 2.4%, if you look on the right, of variants that were not found by run 1, and vice versa. Some variants were found with run 1 that weren't found with run 2, 3.7%. And so there is a coverage issue. And this is fairly recent, so this is relatively the latest technology for sequencing. Again, it's pretty deep. It's deeper than most people get their genome sequenced, which is more like 35x. So the reason for this is that some regions of the genome aren't covered very well for all kinds of reasons. There's all kinds of errors that show up in genome sequences, and so it's really important to understand where they are and where they come. And it's also important to increase coverage in these problematic regions. And I can tell you, I'm going to skip over this next slide because we're getting a little late, but if you do exome sequencing or whole genome sequencing, you will be missing a lot of useful information. And once again, here's a slide from Personalis. There's about 6,000 medically useful genes in the human genome. A normal exome only covers 2,000 of them to 99% coverage at 20-fold coverage. And so you're missing, for most genes, there's a blind spot, if you will, in the exome sequence. And most people don't realize that, but it's true for most, so it's true for 2/3 of the genes. And you can design custom exomes, and this is what my company has done, so again, I am biased here. You can design exomes to backfill these regions and cover them more accurately. And this next slide I won't have time to, but it goes over this in more detail. The blue is the region covered. If you look at these various regions, blue is what gets covered by a normal exome. Green is what you can fill in if you design a specialized exome chip and actually adjust the technologies a little bit. So you can capture these regions, but you do need specialized methods for doing so because of some of the problems that are there. All right, I see I'm running out of time, so I'm going to wrap this up. There are also issues with genome interpretation. I mentioned this telomerase allele. I had this mutation in TERT. That actually predicted I should have aplastic anemia, which I don't. And so we think this mutation is probably contextual, but in some people it causes aplastic anemia, and others it doesn't. And so there are all kinds of issues like that you have to deal with. There are other issues, like most variants that probably are associated with disease don't lie in coding regions, and so we and other groups are working very, very hard to actually now incorporate these into genome annotation. Again, because it's late, I won't go into this, but the top line shows incorporating mutations in protein coding genes into genome annotation. The bottom is the non-coding part. We looked at, if you look in the middle, we zoom in on those variants that affect transcription factor binding, and there are many, many more of them, no matter how you slice it or dice it. There are 10 times more variant-associated alleles that are problematic, that are affecting regulatory sequence than there are damaging alleles for protein coding sequence. And so we think you really do need to get them incorporated. And in fact, there are many cases where you'll have genes with a mutation, and here's one example down below, where you have one mutation in a coding region gene, and the other mutation will be in a regulatory sequence. And so if you only looked at coding, you would miss this. You would think, well, they're heterozygous, it's okay. But it turns out, at least in this genome, the person actually has 2 different mutations that should both be crippling, and you want to collect that information. We're also bringing in other sorts of information like methylome. And lastly, I would say we want to bring in other types of information as well. We're measuring all kinds of activities to bring in so we can start bringing the exposome better. In this case, I would call it the activityome, I guess, where you're following your walking and cycling and all kinds of things. And I'm all wired for these things, as are other people are doing quantified self measurements as well, people like Larry Samar and others. And we're actually bringing this, and the goal is to integrate this information with omics profiling. So it's gotten a little late, so I'll just wrap this up. But personal genome sequencing is here. We think it's not only sequencing, you should bring in other omics information, and you can get a much more detailed perspective of what's going on with the health state. And one of the important things I want to leave is this one down here, number 5, which is every person's disease profile is different. This is the power of personalized medicine. And what we really want to be able to do is follow for every individual. There are changes that occur so you can actually see them early for that individual, not just what it means on average, but for that individual, when their health state changes, you want to catch it and basically prescribe appropriate treatments. And the other thing I'll tell you is because the information is getting so complex, I think you're going to be responsible for your own health. And I think this is an important lesson all of us should take. So here is the data, any of the data we've published. You can find it this website listed at the bottom. And lastly, there's a lot of people who have contributed to this work. I won't have time to go through them all, but it's really spearheaded by Rui Chen, George Mias, Hugo Lam, Jennifer Lee Putan, and Li Hua Jiang. Lots of wonderful collaborators at many different levels. And I do like to thank my mom, who's a great sport in many parts of the study. So I want to thank her, and I'll open this up to questions now. So save time for a little bit of time for questions, so if you have any. Okay. Okay. So I see there are a number of questions here. A lot are in the area of cost. What does it cost to do this? So let me say at the moment that this is very much a research study. We're trying to collect as much information as possible, and we're going as deep as possible to really collect very detailed information. And without the microbiome and without the DNA methylome, the cost for a light version of this is probably a few $1,000s. I'd say on the order of about $2,500 or so. Now with the methylome, with the way we do it, and some of the other technologies, the microbiome, that adds more cost on. But we're doing it fairly deep because we're trying to collect all kinds of information like allele-specific expression. So it could be done at a certain level. I should say that's just the raw cost of doing it. The analysis is incredibly expensive because we're still working out algorithms and ways to do this as well. But I think a light version of this could roll out for a few $1,000s or possibly even less, actually probably for $500 in the not so distant future. But right now we're doing it at the very deluxe detail so we can, again, get as much information. Because the ultimate clinical test, I think, isn't going to be billions of measurements. It's probably going to be a few thousand, few hundred to a few thousand measurements. And that will become much more affordable and targetable. So we just have to know what those few thousand things we want to study are. And we don't know that yet until we do more people and more of these studies. And I forgot to say we are rolling this out to 50 pre-diabetics now and other individuals, as you might guess. Okay, so that's a few, let's see, that's one of the questions. So someone asked, what's the current status of gene profiling patients at risk for progressive kidney disease, patients with type 2 diabetes, and others? And as I say, there's a lot of people doing transcriptomic studies now with these, because that's a very mature technology. The integrative omics profiling, it's only rolling out, as I say, we're rolling it out for type 2 diabetes. We're doing 50 pre-diabetics, people at risk for type 2 diabetes, and we hope to roll in other people. And we are doing some people with kidney transplants and things as well. So we're just starting some of that. So we have, there are many more studies to do, and it does take quite a bit to do one of these studies. They're very, you have to set an infrastructure in place. Actually, that part I forgot to say, that costs millions to set up the infrastructure initially. But now, to execute from that infrastructure is not as bad. So I think it'll roll into these other areas. It'll just take a little bit of time as we move forward, and I know lots of other people we've been teaching how to do this. Here's an interesting question. Someone asked, what is the average data size? How many gigabytes? Well, it's interesting you should say that. It's actually, believe it or not, the first 14 time points we did were 50 terabytes of data we collected on me with the genome sequence and the transcriptome, because we're doing very deep analysis, and the proteome as well can get pretty deep. So we think now with the microbiome and other parts, we're collecting about 6 terabytes currently on every single sample in that range. So about 6 terabytes, and then the genome sequence is another terabyte on top of that. We don't do that every time, but with the methylome and with the microbiome and such, it's about 6 terabytes. So that's a lot. So you can see, you've heard about big data probably, many of you, and this is the ultimate big data. This is literally going to be, well, they're bigger data for astronomy, but for biology, this is quite huge. This will turn out, you can see for me, it's going to be pretty quickly, we'll have a petabyte of data, imagine doing this for a million people, which is what I'd like to see happen. That's 1 million petabytes of data. That gets way up there. So luckily, there are getting to be better and better ways of collecting and storing large data, but we still have a lot of challenges there to go. And it's not cheap to store and manage all that data, I should point out. Someone asked, what is the cost for anyone with all omics interpretations? And that's a difficult one. Until we do all the analyses and things, the final cost, that gets back to the cost one again. So the actual data is probably 2,500. With the microbiome things, it is higher. It's probably 15,000 at the DNA methylome microbiome. But then the analysis on top of that, it does get up there. So I don't have a definitive number, but as these turn into more routine algorithms, they'll get cheaper. Now, someone asked a very interesting question. How do we take into account the impact of epigenetic regulation and when variants in the non-coding region? So I mentioned before the issue about non-coding region variants that hit regulatory sequences that we can now identify. And so we've been bringing in ENCODE and other sorts of data to be able to interpret the regulatory sequence. But this epigenome question is incredibly interesting because we think that's a big part. Again, remember the very first slide that your health is a product of your genome and your exposome, and your epigenome may be reflecting the exposome state. And so we've been bringing the DNA methylome in as well, and I can tell you that's incredibly fascinating. We have a case on one of my alleles where one allele is mutated by mutations, in a typical way. That's my father's allele on a particular gene involved in thyroid function. And interestingly, my mom's allele is actually shut off by DNA methylation as far as we can tell. So that's a complex gene inactivation situation that you would only decipher if you had both types of information. So we're big believers collecting the epigenome is going to be very, very important. Now, in an ideal world, you collect it for different tissues, but we'll just have to collect it where we can, and we can collect it off your iPS cells and things, so don't forget. You can now make iPS cells of yourself, and we've done that for me, that you can do experiments on and see what's going to happen. And so we think between the transcriptome and the proteome and the epigenome, we hope to capture some of that exposome information, even if we don't know the exact exposure one was hit with. So we think that's a certain way to go for this in the future. Is this technology ready for mass application like 23andMe? And the answer is no, not yet. It does require a lot of infrastructure. Just as an amusement aside, we actually did 23andMe after we sequenced my genome. And I was pretty amazed about 2/3 of the predictions did line up between what 23andMe predicted and what my genome predicted and a Toll Butte's group predicted from that. And it was, quantitatively, the numbers weren't the same, but the general concept and idea went the same for about 2/3 of genes. There were some things that we had found that 23andMe missed, but when you think their test is $99 and ours costs several thousand, that's a significant difference. Let's see what else we have here. Let's see, when do you think the next cost step will be, and you can't read the rest of this, for genome sequencing? And yeah, this person's saying it's plateauing. It's a little bit plateauing. I think it's still dropping in price, and as soon as the next competitive technology comes out there, it'll drop quite a bit. And there are some coming down the road. So I predict probably by 2014 you'll see another reasonable drop again. That is a guess. Don't hold me to it, but there are some very interesting technologies coming up, and I think you will see the prices of sequencing dropping even further. There's a goal to hit a $1,000 genome in terms of sequencing. Again, not interpretation. That still costs us about $15,000 to do interpretation the way we do interpretation. That is looking up all your variants. So I'm guessing the $1,000 genome could hit the end of this year, the beginning of next year. That's a guess. It's probably not going to be too far off. Let's see what else we have here. How would you expand the technology using for the autoantibody and the type of significant results obtained? And I think that's a very interesting question. So for the autoantibody stuff, we've been working with a company, Nimblegen, that can actually now display all the human peptides. And so that's how we're now doing it. We're getting better coverage, although it's complementary to what we get with the life technology protearrays. That's how I didn't get into the autoantibody probing, but it's very fascinating. We have cases where we can see the immune system reacting with antigens, and we think that is reflective of disease states. And the nice thing about having your whole virome down on a chip is that you can see the viral probings that appear and the sorts of reactivity you can see. So yeah, so I think the autoantibody stuff is going to be incredibly fascinating. Definitely want to bring this in to the analysis. Someone brought up the issue about privacy and data security, and even brought in National Security Agency, and I guess these are concerns. You know, I think some people are very worried about it. Some people like me aren't. In principle, you're not supposed to be discriminated against by either for employment or for health benefits, but you still can be discriminated against for life insurance or for long-term disability. And I'd certainly like to see the latter go away. But I think, you know, I don't know what to say about the NSA part, but I would argue that the benefits of having your genome sequence, I think, in catching disease early outweighs the concerns about security and people biasing against you. Privacy is an issue. You can figure out someone's identity with reasonable probability with the genome sequence, and as more and more people like me put all their information out there, it'll get easier and easier to link people to their DNA through these public databases, and there's some interesting studies on that already. So, but I would argue the benefits of this are still very, very high, and it's nice to catch this information for benefit. And I think if we had a more protective environment for long-term disability, then I think some of those concerns would go away. I guess I'm not terribly worried about the National Security Agency myself on this, but maybe others are. No, someone asked what are iPS cells, and I forgot. These are essentially stem cells you can make of yourself. You can take your skin and make something called fibroblasts, or your blood, and you can actually turn them into immortalized cells. And in fact, those cells are basically stem cells, and you can grow them in a petri dish, and you can differentiate them into cardiac cells, meaning your heart cells, or nerve cells and various things, and so you can do analyses on these, and they're very, very valuable cells. People someday would like to use these for gene therapy, and there are already trials in place to do this for people who have certain diseases. They think stem cells will be very, very valuable. And I envision a world where most people will have stem cells of themselves, for nothing else than to measure the drug responses and things, so they know what responses they'll be at just from the cells of themselves. I'm not really into cloning oneself is a whole different ballgame that I don't think we're going to be heading down in the near future. So that I don't think we'll have. So I think at this point you should wrap it up. I'm getting the signal to do so. So it's been great chatting online with all of you. If you have further questions, don't hesitate to email me. You can find me by just searching Snyder and Stanford. I think you'll find it. So it's been a lot of fun for me, and I hope you enjoyed it as well. Thanks very much for having me here.